Assalamualaikum warahmatullahi wabarakatuh. My name is ANNUR and today I'm going to present about CRISPR-Cas9. In this presentation, I'm going to talk about what is CRISPR-Cas9, the mechanism of CRISPR-Cas9, its clinical study case, and also the ethical consideration. So, for the introduction, we should know what does CRISPR stand for. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeat. CRISPR is a segment DNA found in bacteria that act like a genetic memory. Cas9 is an enzyme that can cut DNA at precise location. Together, they form a natural defense system that bacteria use to recognize and destroy viral DNA. In biotechnology, this system is adapted to edit gene by guiding Cas9 enzyme to a specific location in genome using a guide RNA or gRNA. This allows scientists to cut, remove, or replace targeted genetic material with high precision. Before we get to know how does CRISPR-Cas9 works, we need to understand a few key components that have in it. The first one is guide RNA or gRNA. It is a custom-made RNA that finds the exact DNA sequence to be edited. The second one is Cas9 enzyme. It cuts both ends strand of DNA at targeted site. And lastly is photospaced adjacent motif PAM sequence, a short DNA tag next to the DNA target site. This required for Cas9 to bind and cut. Next, we will move to the mechanism. The first step is that scientists need to design a guide RNA that matches the faulty gene sequence. The guide RNA searches the genome until it finds a sequence that matches its groups. And for the second step, Cas9 enzyme will only bind if there is a pump sequence right next to the target. This is important to ensure specificity and prevent random cutting or off-target binding. For the third step, this is a cliff step. Once the Cas9 and gRNA complex bind to the target, Cas9 makes a double-stranded break in the DNA. And for the last step, the cells then attempt to repair the break through one of these two pathways. The first one, we have non-homologous end joining, NHEJ, and the second one, we have homology-directed repair, HDR. So what is the difference between these two repair pathways? The first one is non-homologous end joining or NHEJ. This process is fast and does not require a template, but it is error-prone. Small insertion or deletion are often introduced, which can disrupt the gene and essentially switch it off. The second option is homology-directed repair or HDR. This pathway is more precise, but it requires a homologous DNA template. The cell uses the template as a guide to fix and cut accurately, which makes it possible to correct disease-causing mutation or insert entirely new genes. So, in short, NHEG is efficient but less accurate, while the HDR accurately corrects or replaces the DNA sequence. Both are powerful, but HDR is the key for precision therapeutic application. Next, I will be talking about a clinical case study. The case study is about sickle cell disease. So, a sickle cell disease is one of a group of inherited blood disorders. It affects the shape of red blood cells. Normal red blood cells are usually round and flexible. However, in SCD, some red blood cells are shaped like sickle or crescent moons. SCD is caused by change in the gene that tells the body to make hemoglobin. Hemoglobin is the compound in red blood cell that allows the cell to carry oxygen to the rest of our body. The hemoglobin associated with SCD causes red blood cells to become rigid, sticky, and misshapen. For a child to have it, both parents must carry one copy of the sickle cell gene and pass both copies to the child. The symptoms usually appear around 6 months of age. They are vary from person to person and also may change over time. The symptoms include anemia, episode of pain, swelling on both hand and feet, frequent infection, delayed growth or delayed puberty, as well as a vision problem. One of the most groundbreaking applications of CRISPR-Cas9 is Casgevy, the first FDA-approved CRISPR-Cas9 therapy for sickle cell disease. How does it work? Firstly, patient hematopoietic stem cells are harvested and edited outside of the body. Uh, Instead of directly fixing the mutation in the beta globin gene, Casgevy takes a smarter approach, which it edits the gene hematopoietic stem cells to reduce the activity of BCL11A gene. It is a gene that normally shuts off fetal hemoglobin after birth. But by disrupting this enhancer, it then leads to the increased production of gamma globulin and thus higher the level of fetal hemoglobin HbF. HbF has a higher oxygen affinity and prevents the polymerization causing the red blood cell to sickle. Edited stem cells are then reinfused back into the patient and continuously produce the HbF-rich red blood cells. The clinical benefits are remarkable. Patients who once depended on frequent transfusions and suffered from repeated vaso-occlusive crises can now produce their own healthy red blood cells. Casgevy not only reduces the crisis but also avoids the risk of bone marrow transplantation such as graft versus host disease. It is currently approved for patients aged 12 years old and older with recurrent pain crises. Next, we will move to the ethical consideration of CRISPR-Cas9. The first one is germline editing. Editing a heritable DNA is highly controversial because the change can be passed through the future generation. While some bodies, like US national academies, allow its use to prevent severe disease, others argue it should remain prohibited because the concern focuses on the potential of misuse for non-therapeutic genetic enhancement, such as appearance or physical traits, that can lead to a designer baby. The second ethical consideration is the equity and accessibility. Accessibility and distributive justice further complicate the ethical concern. The high costs associated with CRISPR-based therapy like Casgevy raise a concern about societal equity. This scenario raises critical questions about who benefits from this advance and how access can be ensured for the most vulnerable populations. From all that, we can conclude that CRISPR-Cas9 stands as a powerful genome editing tool. It has a great potential in developing therapeutic use. Its ability to precisely edit genetic material has advanced the treatment of previously incurable conditions, as seen in the clinical success of Casgevy for sickle cell disease. However, the technology raises some ethical questions. So, the future of CRISPR depends on balancing the innovation with the responsible use for the benefit of all.